Module A, Clinical Trial Examples. The main goal of this module is to define clinical trial examples that will be used throughout this course. These examples will be very helpful in several respects. First of all, they will be used to help motivate commonly used methods for multiplicity corrections that arise in multiplicity problems that feature combinations of several multiplicity components, such as multiple endpoints, multiple doses or regimens of experimental treatments, multiple patient populations, and also a very important component here, multiple decision points, such as interim analyses or final analyses. And secondly, these clinical trial examples will come in handy when I'm going to illustrate various multiple testing procedures. And throughout this course, I'm going to introduce several numeric examples that we're going to work through to understand the important details of testing algorithms used in multifamily problems. To help us formulate different types of multiplicity problems in this more general and more challenging setting where we have to deal with multivariate multiplicity problems, I'm going to introduce 5 examples. They are listed on this slide as well as the next slide. These 5 examples represent 5 conceptually different settings. For example, example 1 deals with a fairly straightforward problem. It's based on an oncology trial that employs multiple endpoints, which includes primary endpoints and secondary endpoints. Formally, this is a multifamily problem, but in reality, there's only one null hypothesis included within each family, and so we could treat this setting as a problem with a single source of multiplicity. By contrast with this example, the phase 3 clinical trials covered in examples 2 and 3, and these are phase 3 trials in patients with diabetes and schizophrenia, as you can see on the slide. These trials give us examples of problems with 2 sources of multiplicity, which translates into 2 families of hypotheses. But there's also an important difference between these 2 clinical trial examples in that the primary family in example 2, as I will show you, serves as a simple gatekeeper, and in example 3, we will be dealing with a more general type of a gatekeeper. This slide defines 2 more examples. The common feature that we see in these examples is the analysis of multiple clinical objectives at several decision points. As I said a minute ago, examples include a single interim look followed by the final analysis. And this is where things get quite complicated, not to suggest, of course, that the problems arising in the first 3 examples are completely trivial. Here, in example 4, which is based on a prostate cancer trial, a group sequential design will be used, and in example 5, in the breast cancer trial, a more general adaptive design will be considered with options to modify the patient population and also potentially increase the number of patients in the trial. Example 1. Example 1, as I said, presents probably the most straightforward setting that falls under the multivariate multiplicity category. This example is based on a phase 3 trial in patients with prostate cancer. It is loosely based on the all-SIMPCA trial that employed multiple endpoints, primary and secondary endpoints, to provide a better characterization of the efficacy profile of an experimental treatment. Of course, there are numerous other examples of trials with secondary endpoints. I would say that virtually all modern phase 3 trials employ key secondary endpoints for the same main reason. And there was also an interim analysis performed in the all-SIMPCA trial, but we will not going to discuss this aspect of the trial's design. This is a more challenging setting that will be discussed in example 4. So looking very quickly at the information presented on the slide, the objective of this trial was to evaluate the efficacy as well as safety profiles of a novel treatment in patients with castration-resistant prostate cancer, and there was only two arms included in this trial, experimental treatment versus placebo, and therefore this is not a source of multiplicity. Multiplicity was induced in this trial by the analysis of a single primary endpoint and three secondary endpoints. The primary and secondary endpoints are defined as shown on this slide. The primary endpoint is based on overall survival, and it is accompanied here by a set of three secondary endpoints that are defined as the time to first symptomatic skeletal event, then occurrence of new symptomatic pathological bone fractures, and finally occurrence of spinal cord compression. I would like to briefly mention that the role of secondary endpoints in confirmatory trials will be discussed in detail in Module B, and for now I would like to give this quote from Section 6.2 of the EMA's draft guideline on multiplicity issues in clinical trial that was released in 2017. It is emphasized in the guidance document that significant effects in secondary endpoints can be considered for an additional claim only after the primary objective of the clinical trial has been achieved and if they were part of the confirmatory strategy. For this reason, the endpoints that are listed on slide 8 will be tested only if the primary endpoint, which serves as a gatekeeper, is passed, and we can continue to the second stage of the testing algorithm that involves the three predefined secondary endpoints. Example number 2. This example is based on the saxagliptin phase 3 trial in a type 2 diabetes population. The objective of the trial is listed on the slide, and so is the primary endpoint, which was based on the change from baseline to week 24 in hemoglobin A1C. There were also several secondary endpoints investigated in the original trial. They were based on the mean fasting plasma glucose and postprandial glucose, but we'll focus here for simplicity on the primary endpoint. There will be more complex settings presented later on in this module that incorporate secondary endpoints. This saxagliptin phase 3 trial was designed to evaluate the efficacy of several doses and regimens of this novel treatment. Specifically, saxagliptin monotherapy was used as a fixed dose treatment, represented here by the doses of 2.5 milligram and 5 milligrams. And it was also used with a titration approach in the initial treatment of hyperglycemia. Titration of saxagliptin, as you can see, from 2.5 milligrams to 5 milligrams, denoted on the slide by 2.5/5. This was the last treatment arm, and in general, data on titration plays an important role in developing potentially more effective tailoring treatment regimens for patients with type 2 diabetes. And the 3-dose placebo comparisons were grouped as you see on the slide. The comparisons of the fixed doses versus placebo were included in the primary family, and they were followed by the secondary family that included a single comparison. That was comparison of the titration regimen versus placebo. I would like to mention that this is actually a simplified version of the testing strategy compared to the strategy that was employed in this phase 3 trial. And also, as a quick comment, it is often fairly challenging to reconstruct testing strategies used in phase 3 trials. Typically, no diagrams are provided, only simple summaries that may be quite difficult to follow, and therefore, in this online training course, I'm going to put extra emphasis on trying to visualize testing strategies that are used in increasingly more complex multiplicity problems that we encounter in phase 3 trials. Example 3. Moving on to example 3, this phase 3 clinical trial provides an example of a more general multifamily problem. You may remember that an example used in part 1 was also based on a clinical trial from the lurasidone phase 3 development program in schizophrenia. And when I introduced that example in part 1, I stressed that multiple dose placebo comparisons are quite common in phase 3 neuroscience trials, and I added that a phase 3 lurasidone trial in acutely psychotic patients with schizophrenia will be used in part 2 to help illustrate some of the advanced multiplicity adjustment strategies. And there were actually 3 doses of lurasidone studied in that phase 3 trial, and it could have been used as an example in this section. But focusing on the information presented on this slide, the objective of this trial was to evaluate the short-term efficacy and safety of lurasidone in the treatment of acute schizophrenia, and the trial arms included 2 doses of lurasidone, 40 milligrams and 120 milligrams, that were compared to placebo. There was also an active control, olanzapine, which played the supportive role in the original trial. It was included mostly to test for assay sensitivity, and it will not be discussed in the context of this trial example. What would be of more interest of us is to discuss the two-dimensional multiplicity problem that arises in this clinical trial, because the 2 dose placebo comparisons that are defined on slide 14, they define the first source of multiplicity, and this slide shows how the second source of multiplicity was defined. It was defined in terms of 2 clinical endpoints. The primary outcome measure in this 6-week trial was the PANSS total score. PANSS here stands for the Positive and Negative Syndrome Scale. And in addition to that, the trial sponsor pre-specified a key secondary objective based on the Clinical Global Impression Severity, or CGIS score. This CGIS score is computed for a general information from a 7-point scale that requires the clinician to rate the severity of the patient's illness at the time of assessment. And in general, the Clinical Global Impression Rating System includes several scales, including, in addition to this one, the improvement scale. It will be important to add here that there are lots of other examples of multiplicity problems with a very similar structure. In these problems, multivariate multiplicity is induced by the evaluation of several endpoints at multiple doses of an experimental treatment compared to placebo or sometimes to active control. And this slide gives you a very quick example of an extended version, if you will, of example 3, which actually features 3 sources of multiplicity. In this case, the sources of multiplicity were the analysis of multiple endpoints. You can see here on the slide that a single primary and a single secondary endpoints were analyzed in this trial. In addition to that, multiplicity was induced by the evaluation of multiple doses compared to placebo. 2 doses of the experimental treatment were investigated versus placebo. And this slide shows that in addition to those first 2 components, there was another multiplicity component, or another source of multiplicity in this trial. A subpopulation was introduced using relevant genotypic and phenotypic information. This subpopulation, this subset of the overall trial population, was defined based upon pharmacogenetic analysis conducted from prior trials, and the evaluation of the treatment effect in these 2 patient populations, the overall trial population and the predefined subpopulation, defined the third source of multiplicity in this trial. Example 4. The next 2 examples, as I emphasized earlier, are built around clinical trials where multivariate multiplicity was induced by several looks at the data, and in addition to that, standard, if you will, sources of multiplicity, such as multiple endpoints or multiple patient populations. This example is based on the SPARK trial in patients with castrate refractory prostate cancer. And in this case, as you can see, there were two trial arms in the study, and multiplicity was induced in the SPARK trial by the following two factors. First of all, there were two endpoints predefined in the trial. The primary endpoint was based on the time to disease progression, known typically as progression-free survival. The secondary endpoint was based on the evaluation of the treatment effect on overall survival, and those assessments were performed at two pre-specified decision points. There was a single interim analysis employed in the trial, and the final analysis was conducted at the very end of the trial. I think that this example will be very helpful. It will help address questions that I receive virtually every time I give a talk about the analysis of multiple endpoints. I am always asked about performing multiplicity adjustments in oncology trials with several endpoints, such as PFS and OS, that are analyzed at different time points. And we will definitely talk about this important problem. This will be the main topic of Module E. And one last comment. The general principles that are used in the setting, in example 4, can be generalized to phase 3 trials with multiple data-driven decision rules, which includes not only group sequential trials, but also more general adaptive trials. And in this case, this first source of multiplicity may be accompanied by several other sources of multiplicity, such as multiplicity induced by multiple dose control comparisons or several endpoints. Example 5. This last example, example 5, uses certain elements of the BEL-3 trial in patients with locally advanced or metastatic breast cancer, and it includes adaptive population selection. However, this particular adaptive design feature was not actually considered in the BEL-3 study. It will be used here mostly to help us understand the approaches to handling multiplicity in the context of more general adaptive trials. As you can see on the slide, we're going to assume for simplicity the analysis of a single endpoint in the trial, specifically the primary endpoint, which was based on progression-free survival. There were also additional secondary endpoints, such as overall survival or overall response rate, and other endpoints were investigated in the BEL-3 trial, but they will not be dealt with in this example. Instead of that, we're going to focus on the following two sources of multiplicity in this trial. The key components are defined on the slide. We can think of this design as an adaptive population design due to the fact that the interim results could be used to make decisions about the most appropriate patient population to be analyzed at the final look. The predefined patient populations included the overall trial population, the set of patients who met the inclusion-exclusion criteria, and in addition to that, a subpopulation of patients was pre-specified. This was a subpopulation of marker-positive patients. This subpopulation was defined based on a genetic marker based on the PIK3CA gene, and it included patients with gene mutations. In the BEL-3 trial, about half of the patients had mutations of this gene, and the rest had the normal form of the gene. And among the patients with mutations, which means in the subpopulation of marker-positive patients, progression-free survival was expected to be much longer compared to patients in the complementary population. That was the justification behind using a population selection design. I would like to quickly give other examples of trials with adaptive designs. For example, this slide 26, defines a trial that could be thought about as an adaptive treatment selection design. This was a seamless phase 2, phase 3 design in patients with chronic obstructive pulmonary disease or COPD. And the objective of the study was to evaluate the efficacy of multiple doses of an experimental treatment that are defined on the next slide. Those doses were 75 micrograms, 150 micrograms, 300 micrograms, and finally 600 micrograms compared to placebo. The comparison was based on the following primary endpoint. This was the forced expiratory volume in 1 second at 14 days, and multiple decision points were included in the study, specifically a single interim analysis at which a decision could have been made to select only the best doses that would be explored in the second stage of the study, and that would obviously have an important impact on the way the final analysis would be conducted. That final analysis would only deal with the best doses that were chosen at the time of the interim analysis. Wrapping up this module, I would like to clarify that neither the FDA nor EMA multiplicity guidelines provide any information on handling multiplicity issues in adaptive designs. However, what I like about the guidance documents is that they highlight the importance of finding solutions to complex multiplicity problems arising in this context in those challenging adaptive trials. And for example, you can find this quote in the EMA guideline that there are further areas concerning multiplicity in clinical trials, which, according to the above list of issues, are not the focus of this document. For example, there's a rapid advance in methodological richness and complexity regarding interim analysis, with the possibility to stop early due to futility or with a claim for efficacy, or stepwise designed studies with the possibility for adaptive changes in the trial's next steps. We're going to discuss multiplicity issues arising in adaptive designs of this kind in Module F.